If you want a full lecture on CRISPR technology in plants, it's not just a simple explanation, but the whole mechanism, step by step, from start to end, yet in a way that students can easily understand. Here is I have prepared a structured YouTube lecture script so that students and scientists can easily understand how we use CRISPR technology in future. Now, CRISPR in plants, complete mechanism. Now first, I will start. Imagine if farmers could grow rice that never gets fungal disease, or wheat that needs less water, or fruits that stay fresh for weeks. This is not magic. This is a CRISPR technology. Today, let us understand how CRISPR works in plants, step by step, from start to end. It takes me a lot of time to prepare this lecture, so I request you listen it carefully. Maybe it will be fruitful for your future. What is CRISPR? First, we understand what's CRISPR. CRISPR is a tool scientists use to edit genes, like using scissors and glue to cut out mistakes and paste new instructions in plants. How this is possible? This means we can directly change the DNA and controls growth, yield, resistance, and nutrition. My dear, the components of CRISPR. First, we understand what are the components of CRISPR. To understand the mechanism, remember two main players play the part. First, Cas9. Protein, think of it as a pair of sharp molecular scissors. Number 2, guide RNA, gRNA. This is like Google Maps. This is like Google Maps. Listen it carefully. It shows Cas9 exactly where to cut in the DNA. Where to cut in the DNA. That's important step if we use CRISPR in plants. Together, Cas9 plus guide RNA, gRNA, is equal to the CRISPR editing machine. The CRISPR editing machine. Part 3. Step-by-step mechanism in plants. How we use. Number 1. Target identification is very important. Scientists first find the exact gene they want to change. Exact gene they want to change. Example, a gene that makes plants sensitive to drought. Number 2, design of guide RNA. A short RNA sequence is designed to match that gene like a key that fits a lock. This guide RNA, gRNA, will lead Cas9 to the right spot in the DNA. Delivery into the plant cell. This is number 3. The CRISPR system, Cas9 plus guide RNA, gRNA, is introduced. Both are introduced into plant cells. My dear. This can be done. How we can this done? That's important question. This can be done through agrobacterium. agrobacterium-mediated transformation using friendly bacteria to insert CRISPR into plant cells. That is how we can insert in plant cells this CRISPR. Gene gun, biolistics, shooting DNA coated particles into cells. Number 4, that's very important, DNA cutting. Once inside gRNA, guides Cas9 to the target gene. Cas9 makes a precise cut in the DNA. Na udhar, na idhar. Precise cut in the DNA, like scissors cutting a string. Number 5, DNA repair by the cell. The plant's natural DNA repair system now activates non-homologous and joining NHEJ DNA rejoins, but may create small changes, mutations. This is useful for knocking out or disabling a gene. Homologo-directed repair, HDR. Scientists provide a new DNA template and the plant copies it into the cut site. This allows insertion of beneficial traits. Plant regeneration, that's important. Edited cells are grown into whole plants using tissue culture. Every cell of the new plant carries the edited gene. My dear. I hope so that you understand still what I am saying. Now part 4, applications in plants. What are the applications of CRISPR in plants? Disease resistance, bananas resistant to viruses, rice resistant to blight, citrus tolerance, wheat that survives nowadays, drought, rice that grows in salty soils, nutritional improvement, tomatoes with more antioxidants, rice with more vitamin A, yield improvement, faster growing, higher productivity crops. Ethical, that's a part 5, ethical and safety concern, that's very important. CRISPR is powerful, but we must be careful. Will edited plants affect biodiversity? That's very important. How will they be regulated as GMOs, genetically modified, or non-GMOs? These are important questions for scientists and governments. These are very important concerns which are connected with CRISPR technology. Call to action, my dear. So dear students, scientists, CRISPR is like giving us a pen to rewrite the book of life. In plants, it can solve hunger. Malnutrition, and food security. That's very burning issue around the world because the population is going to double up to 2050. These are the challenges. But remember, powerful science demands responsible minds. The future of agriculture lies in the hands of students and scientists like you. You will use CRISPR not just to feed people, not just to feed people, but to nourish the plants, but to nourish the plants, but to nourish the plants. Me, Ahmad Mushtaq, creator of Kalima Foundation Kashmir, I hope so that you like and share and subscribe my channel.